PCR stands for polymerase chain reaction. The polymerase being referred to is the DNA polymerase used to elongate the target strand in the 5' to 3' direction. In this case, we are looking at TAQ DNA polymerase specifically. TAQ polymerase was derived from the Thermus aquaticus bacteria found in Yellowstone National Park in 1988. This polymerase is used because it is stable at the high temperatures used in the PCR process. This chain reaction facilitates the rapid production of copies of a specific target strand of DNA for testing by cycling through 3 different temperatures. The 4 nucleotides needed to create these copies, A, T, C, and G, are found in the replication medium.
PCR amplification happens in 3 steps. The first step is denaturation, in which the thermocycler heats the DNA to 90 to 100 degrees Celsius and separates the strands of DNA the way DNA helicase does in normal DNA replication. The second step is annealing, which the thermocycler cools to 30 to 60 degrees, and it allows short single-stranded primers to attach to the 3' end of each DNA strand. The last step is synthesis, and for TAQ DNA polymerase, it heats to about 72 degrees Celsius and elongates the DNA in the 5' to 3' direction. Now these 3 steps make one cycle, and the cycle repeats with each new strand for exponential amplification. The formula to calculate how many segments of DNA you have after a couple PCR cycles is x, where x is the number of segments you started with, times 2 to the n, and n is the number of cycles because each time you double the number of DNA segments.
Before we begin discussing the applications of PCR, it is important to understand its limitations. First off, prior knowledge is required in order to construct the necessary primers. Secondly, with PCR's ability to modify extremely small amounts of DNA, any minute amounts of DNA from lab workers, such as flakes of skin, has the ability to contaminate the sample. A third limitation of PCR is that the polymerase used, TAQ, does not have the ability to proofread, unlike other DNA polymerases, so an incorrect nucleotide is incorporated about once every 20,000 base pairs. Finally, the size of the fragments that can be amplified by standard TAQ polymerase is usually less than 20,000 base pairs, and even with modifications, the length is limited to 50,000. Despite these limitations, PCR has become a widely used tool. For example, PCR can be used to detect the presence of viruses in blood samples by adding primers complementary to known viral DNA sequences. For the virus's presence, the primer will attach to DNA containing the virus, resulting in the amplification of a DNA fragment of a known length. Therefore, the presence of a DNA fragment of that length on a gel indicates the presence of viral DNA in the tested blood sample. Modern tests for HIV commonly use this process.
Let's see what you remember with this pop quiz. What temperature could annealing occur at? 42 degrees Celsius. What is the formula for the exponential amplification of DNA with one original fragment? 2 raised to the number of cycles. Which of these is an application of PCR? Both A and B.